there's this family of enzymes called sirtuins. And the first one that was discovered, SIRT1, was associated with enhanced longevity. And that work was actually done in worms. C. elegans, a worm model, where they knocked out the gene and it shortened lifespan, and they increased SIRT1 and it extends lifespan. And then a family of related genes were cloned or identified. One of them is SIRT3, and SIRT3 is localized in mitochondria, but nowhere else in the cell. The SIRT1 is actually in the nucleus that regulates gene expression. But SIRT3 is an enzyme in the mitochondria, and what it does is it removes acetyl groups from lysine residues in proteins. Some of the viewers may know about protein phosphorylation, where a phosphate group is added to certain amino acid residues in proteins, serine, for example, or threonine, or tyrosine. And then, so there's enzymes that add the phosphate and enzymes that remove the phosphate, so kinases and phosphatases. So with protein acetylation, it's similar. There's enzymes that add an acetyl group to a protein. They're called acetyltransferases. And there's enzymes that remove the acetyl group. They're acetylases. SIRT3 is an acetylase. And there's a number of interesting proteins that are deacetylated by SIRT3 in mitochondria. One is the most important antioxidant enzyme in cells. It's called superoxide dismutase 2, SOD2. And the deacetylation by SIRT3 enhances the antioxidant enzyme activity of SOD2. So anyway, in that way, that SIRT2 and 3 helps remove free radicals from the mitochondria after they're produced. Another protein that we studied that's deacetylated is a protein called cyclophilin, and it's involved in regulating a process that triggers cell death or apoptosis. And the deacetylation of that protein will protect the cells against cell death. So anyway, we collaborated with another group. This is when I was a lab chief at National Institute on Aging. And we collaborated with a group actually at National Cancer Institute, and they made SIRT3 knockout mice, and then we characterized essentially the brains of these animals. Actually, they seem pretty normal, but as they get older, they start to have problems. There's problems with hyperexcitability of neural networks, which we found by EEG recordings, but we didn't see any neural death or obvious degeneration of neurons. So we crossbred the SIRT2 and 3 knockout mice with a mouse model of Alzheimer's disease, and we found that there's a dramatic acceleration of neural hyperexcitability and cognitive deficits and amyloid and tau pathology in the animals that have reduced levels of SIRT3. So that suggests that this protein in mitochondria has important roles, and interestingly, in regulating neural excitability and vulnerability of neurons to aging and Alzheimer's disease, at least in these kind of preclinical studies.